Good evening, everybody. So let's start our lesson. Today, the microbes in human welfare. The another important chapter in our term 2 syllabus. So before starting this chapter, we can say that pest infestation in food crops has been banned for ensuring food security across the various parts of the world, destroying lakhs of tons of food crops like seeds, aphids, and food borer every year. Several institutions across the globe have gradually developed pest-resistant crop varieties using targeted plant breeding experiments. Indian Agricultural Research Institute, the IARI, PUSA, has also developed a number of crop varieties pertaining to pest resistance in fruits and vegetables. For example, the Pusa swani, which is a resistant to shoot and fruit borers. So starting to our new topic. In our syllabus, the subtopics are the microbes in food processing and industrial production, sewage treatment, energy generation, microbes as biocontrol agent and biofertilizers, antibiotics, its production and its judicious use. As usual, the marked description. This is chapter number 10. We already completed chapter 8 and chapter 10. Both are collectively give us 14 marks. So we are preparing for 14 marks. And the biotechnology, that is 11 marks. And ecology and environment, that 10 marks. The 34 marks examination. So these are the different important pictures in our NCERT. So go through NCERT and make sure the diagram or keep it into mind. So that's the rod shaped. Rod shaped. These are the spherical shaped, and this is the structure of a rod shaped bacterium and its flagella. And here the well known bacteriophage, that's very important, we know this one. The head, the collar, the tail, the plate, the pins and the prongs are all shown here. This is the adenovirus causing the respiratory infection, and the third diagram, that is the rod shaped TMV, the tobacco mosaic virus, are shown here. So come in contact with this diagram because sometime from there some questions also asked. The next diagram that is the colonies of bacteria. This is the petri dish and the colonies of bacteria are there. And in this petri dish, the fungal colony are shown. Now the main topic, the microbes in household products. The common example in the production, that is curd from milk. The microorganism such as lactobacillus and other commonly these are called lactic acid bacteria, LAB. They grow in milk and convert it to curd. This is our household products. During growth, the lactic acid bacteria produce acids that coagulate and partially digest the milk proteins. A small amount of curd added to the fresh milk as inoculum or a starter that contain millions of lactic acid bacteria, which is suitable temperatures, multiply, thus converting milk to curd, which also improves its nutritional quality. So remember this one, the curd is more nutritious than milk because it increasing some vitamins like vitamin B12. In our stomach too, lactic acid bacteria play a beneficial role in checking disease causing microbes, and the dough which is used for making the foods such as dosa and idli is also fermented by the bacteria. Dough is a puffed up appearance, and this puffed up appearance is caused due to production of CO2 gas. That's very important. Similarly, the dough which is used for making bread is fermented using baker's yeast. You know this one, the scientific name, baker's yeast, the Saccharomyces cerevisiae. Frequently asked question, what is baker's yeast? A number of traditional drinks and foods are also made from fermented by the microbes. So these are the some use in household products. Toddy, another household product, traditional drink, some parts of the southern India. Microbes are also used to ferment fish, soybean and bamboo shoots to make food. Cheese is one of the oldest food items in which the microbes are used. Different varieties of cheese are known by the characteristics on the basis of their texture, flavor, taste, thus specifying they coming from the microbes used. For example, one of the important topic of today, Swiss cheese, are due to the production of large amount of carbon dioxide by a bacterium, Propionibacterium shermani. Keep it into mind. And here is the Swiss cheese in front of you. The holes are making by the carbon dioxide. The Roquefort cheese are ripened by growing a specific fungi on them which give them a particular flavor. So moving from the household to the industry, in industrial products, even in industry, microbes are used to synthesize number of products valuable to human being. Beverages and antibiotics are some example. Production in an industrial scales require growing microbes in very large vessel. So these are the fermenters. These large vessels are used as a fermenter, and this is a fermentation plant. Now the fermented tanks, the fermented beverages are produced. Here the microbes are specially yeasts, been used for a time immemorial for the production of beverages like wine, beer, whiskey, brandy or rum. For this purpose, the Saccharomyces cerevisiae, the same yeast, is used for bread making and commonly called brewer's yeast. So baker's yeast and brewer's yeast is used for fermenting malted cereals and fruit juices to produce ethanol. Very important. Depending on the shape, the raw material used for fermentation and the type of processing with or without distillation, different types of alcoholic drinks are produced. Wine and beer, very important, keep it into mind, produced without distillation. So wine and beer are produced without distillation, whereas whiskey, brandy, rum are produced by distillation in the fermented broth. That's very important. So keep it into mind, wine and beer are without distillation, and whiskey, brandy and rum are distillation. These are the distillation product. Now in our next topic that we discussed in the previous pages, the different types of alcohol. Now the alcohol and their substances used and their alcoholic concentration in this group. So here you see beer. So previously we discussed these are the no distillation, the substances item, the barley, and the alcoholic concentration, that is 3 to 6%. Wine, that is also no distillation product. Wine always from fruit juice, and alcoholic concentration, that is 9 to 12%. These are all distillation product. These are all distillation product. Whiskey from malted cereals, brandy from fruit juice, rum from molasses, and vodka from potato. Very important. In whiskey, the alcoholic concentration is 50%. In brandy, 60 to 70%. Molasses, the 40%. And the vodka, that is from 60 to 80%. So percentage are to be remembered because some questions are asked also from here. And that's very important, which are distillation product, non-distillation product, and the substances from the alcohol are produced. So moving to the next page. Antibiotics produced by microbes are regarded as one of the most significant discoveries of the 20th century and have greatly contributed towards the welfare of human society. Anti is a Greek word that means the against, and bio means life. Together they mean against life. That means disease causing organisms, whereas with reference to human being, they are pro-life and not against. Antibiotics are the chemical substance which are produced by some microbes and can kill to retard the growth of the other disease-causing microbes. Alexander Fleming, well known to all of you, while working a Staphylococci bacteria, once observed the mold growing in one unwashed culture plates, which is Staphylococci, would not grow. He found out of this it was due to chemical produced by the mold, and he named it penicillin after the mold, Penicillium notatum. However, its full potential is an effective antibiotic was established much later by Ernest Chen and Howard Florey. This antibiotic was extensively used to treat American soldiers wounded in World War II. Fleming, Chen, Florey were awarded Nobel Prize in 1945 for their discovery. We all know Alexander Fleming, but beside this, we have to remember Ernest Chen and Howard Florey. Fleming, Chen and Florey. After penicillin, other antibiotics were also purified from other microbes. Antibiotics have greatly improved our capacity to treat with deadly diseases such as plague, whooping cough, diphtheria, leprosy, which used to kill millions all over the globe. But after antibiotic, today, this type of disease cannot destroy our life. So today we cannot imagine the world without antibiotics. Now the chemicals, enzymes and other bioactive molecules. Microbes are also used for commercial and industrial production of certain chemicals like organic acid, alcohol and enzymes. Examples of acid producers and Aspergillus niger, very important. The fungus of citric acid, Aspergillus niger, Acetobacter aceti of acetic acid, Clostridium butyricum, butyric acid, Lactobacillus, lactic acid. Yeast is used for commercial production of ethanol. So very important, citric acid produced product, from where the citric acid are produced, from where the acetic acid are produced, and from where the butyric acid are produced. You can remember the Clostridium butyricum, then there is a resemblance with butyricum and butyric acid. Acetobacter aceti, from acetic to acetic acid, but the most important is Aspergillus niger, because there is no resemblance with the scientific name with the produced acid. So that's very important. Citric acid is produced from Aspergillus niger. Microbes are also used for production of enzyme. Lipases used as detergent formulation that are helpful to removing oily stains from the laundry. Very important. Lipases. Bottled fruit juices brought from the market and clear and compared to those made at home. This is because the bottled juices are clarified by the use of pectinases and proteases. Very important. The two enzymes, pectinases and proteases, are used that clarified the bottled juices. Streptokinase, very important topic. Streptokinase, produced by the bacterium Streptococcus and modified by genetic engineering as clot busters. For removing the clots, sometimes we are facing the problem of blood clotting. So clots from the blood vessels of the patient who have undergone myocardial infarction leading to the heart attack. So this drug, so very important component, keep it into mind, streptokinase, that helps us from the myocardial infarction that led to the heart attack. Another bioactive molecule, cyclosporin A, that is used as an immunosuppressive agent in organ transplant patient. Very important. Produced by the fungus Trichoderma polysporum. One of the important question. Trichoderma polysporum. Another important, that is statins, that produced from the yeast Monascus purpureus. Monascus purpureus, that has been commercially as blood cholesterol lowering agents. So cyclosporin A and statins, they are frequently asked questions. It acts as a competitively inhibiting the enzyme responsible for synthesis of cholesterol. So look at this one, how important it is. Now, our next topic, microbes in sewage treatment. So look at here, the raw sewage. After screening, the collecting tank is there, then it moves to the primary settling tank, then the aeration tank, from aeration tank the secondary settling tank, now colonization system, the chlorine are come in contact with this tank. This is the another filter, and now the treated water for ready to reuse. And this from this primary treatment and the secondary treatment tank, the sludge drying beds are used for disposal. So it's the easy chart of sewage treatment process. So take a screenshot for further reference. Now the first page, the primary treatment. The treatment steps basically involve the physical removal of particles, large and small, from the sewage through filtration and sedimentation. These are removed in stages. Initially, floating debris removed by sequential filtration. Then the dirt, the soil and small pebbles are removed by sedimentation. All solids that are settled from the primary sludge and the supernatant forms the effluent. The effluent from the primary settling tank is taken into secondary treatment. So the components which are moving from primary to the secondary, these are effluent. So that's the diagram to explain the primary treatment. As a short note, what is primary treatment? So after primary treatment, the secondary treatment. Look at here, the primary effluent is passed into a large aeration tank, where is the constantly agitated mechanically air is pumped into it. This allows vigorous growth of the useful aerobic microbes in presence of oxygen into flocks. So that's the important question. What is flocks? So flocks are the masses of bacteria associated with fungal filament in form of mesh-like structures. So very important this one. What is flocks? While growing, the microbes consume the major part of the organic matter is effluent. This significantly reduces the BOD, biological oxygen demand, of the effluent. BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. The sewage water is treated till the BOD is reduced. The BOD test measures the rate of uptake oxygen by microorganisms in a sample of water, and thus indirectly BOD is a measure of organic matter present in water. The greater the BOD of wastewater, more its polluting potential. Very important line. Once the BOD of sewage or wastewater is reduced significantly, the effluent is then passed into a settling tank where the bacterial flocks are allowed to sediment. This sediment is called activated sludge. Very important. What is activated sludge? A small part of activated sludge is pumped back into the aeration tank to serve as the inoculum. The remaining major part of the sludge is pumped into the large tank called the anaerobic sludge digesters. Here other kinds of bacteria which grow anaerobically digest the bacteria and the fungi in the sludge. During this digestion, the bacteria produce a mixture of gases such as methane, hydrogen sulfide and carbon dioxide. So these are the components. It's a very important question. What are the mixture of the gases and what are the gases present in this mixture? These gases from biogas and can used as a source of energy. It is inflammable. The effluent from the secondary treatment plant is generally released to the natural water bodies like rivers and streams. You can appreciate how microbes play a major role in treating the millions of gallons of wastewater every day across the globe. So very important one. This methodology has been practiced for more than a century now in almost all parts of the world. Till date, no man-made technology has been able to rival the microbial treatment of sewage. You are aware there that due to increasing urbanization, we all aware about this one, sewage is being produced much larger quantities than ever before. However, the number of sewage treatment plants has not increased enough to treat such large quantities. So the untreated sewage is often discharged directly into rivers, leading to their pollution and increase in waterborne diseases. The Ministry of Environment and Forest has initiated, that is GAP, Ganga Action Plan, and YAP, Yamuna Action Plan, to save these major rivers of our country from pollution. Under these plans, it is proposed to build a large number of sewage treatment plants so that they treated sewage only by discharge in the rivers. A visit to sewage treatment plant situated in any place near you would be a very interesting and educating experience. So those are living in Chittaranjan, you on the way of BRS, you are getting this type of treatment plant, and after taking permission, you are permitted to observe this one. So that's the practical experience how this one is going to form. Now the next topic, microbes in production of biogas. So this is a typical biogas plant. Frequently asked question from this one. This is the dung and water are deposited here. This is the gas holder. This is the digester, and after this the sludge is collected there. So everybody take a screenshot for drawing this one. Biogas is a mixture of gases containing predominantly methane produced by the microbial activity and which may be used as fuel. You have learned that microbes produce different types of gaseous end product during growth and metabolism. The type of gas produced depend upon the microbes and the organic substrate they utilize. In the examples cited in relation to fermentation, dough, cheese making and production of beverages, and main gas produced was CO2. However, certain bacteria which grow anaerobically on cellulosic material produce large amount of methane along with carbon dioxide and hydrogen. These bacteria are collectively called methanogens, that is methanobacterium. These bacteria are commonly found in the anaerobic sludge during sewage treatment. The bacteria are also present in the rumen, very important. It's a part of the stomach of cattle. A lot of cellulosic material present in the food of cattle are also present in the rumen. In rumen, these bacteria help in the breakdown of cellulose and play an important role in the nutrition of cattle. The biogas plant consists of a concrete tank 10 to 15 feet deep in which the biogases are collected and the slurry of dung is fed. The floating cover is placed over the slurry which keeps on rising as a gas in production in the tank due to the microbial activity. The biogas plant has an outlet which is connected to a pipe to supply biogas to nearby houses. The spent slurry to removed through another outlet and may be used as fertilizer. Cattle dung is available in large quantities in rural areas where cattle are used for a variety of purposes. So biogas plants are more often built in rural areas. The biogas thus produced is used for cooking as well as lighting. The technology of biogas production was developed in India due to the effort of IARI and KVIC, very important abbreviation, Indian Agricultural Research Institute and Khadi and Village Industries Commission. So very important to remember this abbreviation, IARI and KVIC. If your school is situated in a village near a village, it would be very interesting to inquire if there are any biogas plants in nearby. You can observe this one practically to get the proper knowledge. Moving to our next part, that is the microbes as biocontrol agents. Biocontrol agent, these are biocontrol refers to the use of biological methods for controlling plant disease and pests. In modern society, these problems have been tracked, increasing the use of the chemicals by use of insecticides and pesticides. These chemicals are toxic and extremely harmful to human being and animal alike and have been polluting our environment, soil, groundwater, vegetables and crop plants. Our soil is also polluted through our use of weedicides to remove weeds. Biological control of pests and diseases. In agriculture, there is a method of controlling pests that relies on natural predation rather than use chemicals. A key brief of the organic farmer is that biodiversity further sells. The more variety of landscape has, more suitabilities. The organic farmer therefore work to create a system where the insects that are sometimes called pests are not eradicated but instead are kept manageable level by complex system of check and balances within living and vibrant ecosystem. So this is a very important part of our chapter. Everybody must give your response and share the class to among your friends. Very few are listening now. I want a lot of groups that are listening now. So come and join the lesson. Contrary to the conventional farming practices, which often use chemical method to kill both useful and harmful life forms indiscriminately. This is the holistic approach that seeks to develop understanding of the waves of the interaction between the myriad of organisms and constitute the field, fauna and flora. The organic farmer holds the view that the eradication of the creatures that are often described as pests is not possible, but also undesirable, for without them the beneficial predatory and parasitic insects which depend on them as food or host would not be able to survive. Thus the use of biocontrol measures will greatly reduce the dependence on toxic chemicals and pesticides. That's the utility. An important part of the biological farming approach to become familiar with the various life forms that inhabit the field, predators as well as pests, and also their life cycle, patterns of feeding and the habitats that they prefer. This will be developed appropriate means of biocontrol. The very familiar beetle that is red and black marking. I think you all know this one and observe this one. The ladybird and the dragonflies are useful to get rid of aphids and mosquitoes. Respectively, an example of microbial biocontrol agent can be introduced in order to control butterfly caterpillars in the bacteria Bacillus thuringiensis, often called BT. These are available in sachet or dried spores which are mixed with water and sprayed onto the vulnerable plants such as brassicas and fruit trees, where they are eaten by the insect larvae. In the gut of the larvae, the toxin released and the larvae get killed. The bacterial disease will kill the caterpillars but leave other insects unharmed. Because of the development of methods of genetic engineering, in the last decade or so, the scientists have introduced Bacillus thuringiensis toxin genes into plants. Such plants are resistant to attack by insect pests. BT cotton, the one such example, which is often being cultivated in some states in our country. The biological control being developed for use of the treatment of plant disease in the fungus Trichoderma, very important. Trichoderma species are free-living fungi that may very common in the root ecosystems. From there, we are moving to the next. They are effective biocontrol agents for several plant pathogens. Baculoviruses are the pathogens that attack insects and other arthropods. Very important and frequently asked question, baculovirus are attack the insects and the other arthropods. The majority of baculovirus used as biological control agent and are the genus NPV. That is the NPV nucleopolyhedrovirus. These viruses are excellent candidates for species-specific, narrow spectrum insecticidal applications. So they have been shown to have no negative impact on plants, mammals, birds, fish, and even non-target insects. So it is a eco-friendly component. This is especially desirable when beneficial insects are being conserved to aid an overall integrated pest management program or when the ecologically sensitive area is being treated. Next topic, that is the microbes and biofertilizer. With our present day life, styles environment pollution is a major cause of concern. The use of chemical fertilizer to meet the ever increasing demand of agricultural produce has contributed significantly to this pollution. Of course, we have now realized that there are problems associated with the overuse of chemical fertilizers, and there is a large pressure to switch to organic farming. The use of biofertilizers. Biofertilizers are organisms that enrich the nutrient quality of the soil. The main sources of biofertilizers are bacteria, fungi, cyanobacteria, and these bacteria fix atmospheric nitrogen into organic forms, which are used by the plant as nutrient. Other bacteria can fix atmospheric nitrogen while free living in the soil, that is Azospirillum and azotobacter. Remember this one. These are free living nitrogen fixing bacteria, thus enriching the nitrogen content of the soil. Fungi are also known from symbiotic association, that is the mycorrhizal association. Many members of this genus glomus from mycorrhiza. The fungal symbiont, you all know the symbionts is. Anyone can tell me what is symbiont? Can anybody tell me what is symbiosis? So symbionts, these are the association. Yes, Ankita, you are right. The mutual interaction, and the symbol is plus plus. Their association absorbs phosphorus from soil and passes it to the plant. Plants having such associations show other benefits also, such as resistance to root-borne pathogens, tolerance to salinity and drought, and an overall increase in plant growth and development. Cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environment, many of which can fix atmospheric nitrogen, that is Anabina, Nostoc and Oscillatoria. In paddy field, that's very important, cyanobacteria serve as an important biofertilizer. The blue-green algae also add the organic matter to the soil and increase the fertility. Currently in our country, a number of biofertilizers are available commercially in the market. So that is going popular nowadays. And farmers are use them regularly in the fields to replenish the soil nutrient, to reduce the dependence on the chemical fertilizers. So good things are yet to come. Lot of things are yet to come. So now, moving to our last part, see this one. This is your mind map to remember the chapter at a glance. So the industrial use of microbes, sewage treatment, primary, secondary, tertiary, biofertilizer, biocontrol agent, biogas production, household product, energy production. So remember this one. And the Bacillus thuringiensis, the cry gene, all these are there for ready for your mind map after the completion of this chapter. So I think you all revise with me the whole chapter of microbes in human welfare. That will be helpful for your turn to exam. So till then, thank you everybody. We meet in the next class with our next chapter tomorrow. That is biotechnology. The another important part of our turn 2. So bye everybody. Good night.